Hello everyone, myself Dr. Silpi Sarkar, native of Bahrampur, West Bengal, and I am working as an assistant professor in Vignan's Foundation for Science, Research and Technology in the Department of Biotechnology in Andhra Pradesh. So during my PhD, I worked on freshwater snails, land snails, and tried to determine the morphology of the snails, and also I tried to understand the molecular phylogeny of these conspecies of these snails related to different places of India. So during my PhD, I learned the techniques such as DNA isolation, PCR, agarose gel electrophoresis, then SDS page, morphological observation by Excel software, and also the different techniques like ELISA and Western blot. Also, I have gone vividly to different conferences and international conferences where I presented my research paper during my PhD. So, all through my PhD, I have done active research on snails and published a few research articles in the SCI and Scopus journals. I have also attended several conferences and workshops during my PhD. So, also, other than my research and review papers, I have also extensively researched and written certain book chapters, and also I have been the editor of a e-magazine in known as Times Tech India, and I am working as a voluntary editor there. So this is a brief review of my PhD and my research and my place where I used to stay. I am personally very glad that Dr. Karan Singh has invited me to give a talk and inspire the students who are currently hearing my talk. So, as we all know that science is always has to be searched through literature. Science is practical. Science can be challenged. So science is the itself a subject which has its logic, and also it has its own potential. So being a scientist and being in the progress of science itself is very cherished dream of everyone, and I hope everyone will always try to find the fact and the logic behind anything. During my PhD, I was always fascinated to understand the biological concept of species, the whole study of the taxonomy, the classification of naming and grouping the species and the organisms. So, when I took up the challenge of the molecular phylogeny of the snails, which was not much done within the eastern India, so my first thought was, let me go to the literature and check the facts, what is the thing done, what are the research work done. So I found it very interesting that most of the research was done was to identify a snail species or to identify a name a snail species. Much molecular biology or DNA recombinant work was not done. So in that lacuna I find that okay, let me try out on these. Thus, while my interest developed slowly, I took up the invertebrate study, that is the animal study. Normally people will go for medical or the vertebrate study of science. But this place of invertebrate was lacking in India. So I started my study, and then to understand deeper, I tried to understand the science of taxonomy. So thus, the science of taxonomy deals with the systematic study. So systematics has had a remarkable renaissance in recent generations. Now taxonomists who study taxonomy is a scientific study of naming, defining, and classifying the groups of biological organisms which are based on shared characters. So, when I tell about shared characters, we must understand that characters of each and every organism has some similarity with its ancestor. So, to understand the similarity of the characters, to find out the similarity or the matching identity between different characters, we try out with a kind of softwares which helps to understand the ancestor or evolutionary relationship of one species with the other. So this is the study of molecular phylogeny. To understand the molecular phylogeny, we need to understand the taxonomy behind a naming of a species. Thus, taxonomy has been studied. So taxonomy study has been done by experimental method and comparative method. So whenever I say about comparative method, we need to understand that comparison is always based on observation. So there can be two branches of taxonomy. One is microtaxonomy at the species level. Another is macrotaxonomy, that is classification at the higher level. So according to the book of Principles of Systematic Zoology by Mayer and Ashlock, more than 1 million species of animals and 500,000 species of plants and microorganisms have been described and estimated over the number of living species range from 3 million to 10 million and even higher. Now each species can be discriminated based on its different forms such as sex, age, class and seasonal forms. The term taxonomy is derived from the Greek word taxis, that is arrangement, and nomos means law, and was first proposed in its French form by de Candolle in 1813 for the theory of plant classification. Now the term systematics stems from the Latinized Greek word systema, given by Linnaeus. Thus, systematics can be defined by the study of its different kinds and the diversity of its organisms on any or within the relationship among other species. Now, systematic deals with population, species, organism, its higher taxa, levels of classifications, and all this. So, what is the use of studying all this taxonomy, species, biological diversity, all this? There is certain uses. So let us go to the applicative part. First application is to determine and compare the unique properties of each species within itself and also with its higher taxa. Second, to determine what properties certain taxa have in common and what are the biological causes of the differences or the shared characters. Thus, multiple role of taxonomy in biology can be summarized as follows. It is the only science that provides a vivid picture of the existing organic diversity of the earth. It provides most of the information needed for a reconstruction of the phylogeny of life. It simplifies almost exclusively the information needed for entire branches of biology. Example, biogeography, zoogeography, and the geography of the evolutionary organisms. Now, the diversity of the living world is one of the most interesting and challenging aspects of nature. Some organisms are haploid, deriving most of their life cycles, while others are diploid. The first and the most basic task of taxonomies is to sort the bewildering diversity of individuals which are found in nature. The activities by which these achieved constitute the microtaxonomy. The terminologies which we need to understand to study systematics are A. Phenon. This is used to describe the different forms or phenotypes that may occur within a single population. B. Taxon. Defined by Simpson in 1961. It is used as a group of real organisms recognized as a formal unit at any level of hierarchic classification. The species is not a taxon but a category. Example, in the robin bird, Turdus migratorius is a taxon. Taxa are also known as historical groups. C. That is category. It designates a rank or level in a hierarchy Classification. It is a class where members of all the taxa are assigned a given rank. Terminologies such as species, genus, family, and order designate the categories. Now, once we understand the different terminologies used in the systematic study, let us go into the species concept. So, if we go back into the literature reports innumerable species concept. There are a number of species concepts are there. Among that, 4 species concepts are very much studied. One is typological species concept, second is nominalistic species concept, third is biological species concept, and fourth is evolutionary species concept. Now the typological species concept. According to this concept, the observed diversity of the universe reflects the existence of a limited number of underlying universals or types. Individuals don't stand in any special relationship to each other, being merely expressions of the same. Also known as essentialism concept. This is also known as essentialistic species concept. Second is nominalistic species concept. Here, the nominalist accepts the individuals exist while species are abstractions created by people. This concept is very popular in France in 18th century. Besse in 1908 expressed this point of view that nature produces individuals and nothing more. Species have no actual existence in nature. They are mental concepts and nothing more than that. Species have been invented in order that we may refer to a greater number of individuals collectively. The third concept is biological species concept. The members of a species of animals forms a reproductive community. The individuals of a species of animals recognize each other as potential mates and seek each other for the purpose of reproduction. The series is known as ecological unit, and the species also is known as genetic unit. Can be defined as a group of interbreeding natural population which is reproductively isolated from each such groups. The fourth concept is evolutionary species concept. Simpson in 1961 proposed an evolutionary species is a lineage, an ancestral descendant sequence of populations which evolve separately from others and with its own unitary evolutionary role and tendencies. So thus, going to the literature and studying in the species concept gives us an idea of the taxonomy and the species. So on a whole, this topic is very concisely said here. Vast number of literature, surveys and book chapters are to be read to understand in a vivid way. Hope this topic discussion is little bit useful to all of the people who are much interested in biology, in the species concept, in the taxonomy of classifying or naming organisms, and in the molecular phylogeny. Thank you. Hope you have a wonderful day.